So thank you, everybody, for being here today. Thank you for the invitation to speak. I'm really excited to share with you some of the work that we're doing. Is the mic okay back there? Yeah, okay. The title of my talk, I decided today, would be Plastic in Today's World, and I think you'll see why, if it works. Okay. Oh, I skipped one. Okay, so just quickly, I know Carol Ann encouraged me to say a little bit about who I am personally, in addition to my professional self, and so I just wanted to touch on that a little bit here. So I am a wife, a mom, a friend, family member, etc. I have two kids. They're both boys. One is a freshman and one is a fifth grader. We have two pets. And my husband, Ben Davis, also works the laboratory. I really like getting outside. Sometimes we're dragging the kids along with us these days, but nonetheless, we like to hike. I love paddleboarding. We enjoy skiing, especially when there's better snow. And so as you can see, we have one giant dog and one regular-sized cat. And overall, I love the outdoors, I love nature, and for me, I have a really strong desire to try to make a positive impact on our world. At work, at the laboratory, I am a group leader in Bioscience Division, and the name of my group is Microbial and Biome Sciences. So we mostly focused on how single-celled organisms, small microbes, interact in their natural world, as well as how we can engineer or manipulate them in ways to better our future as a society, as well as for the Earth. And so my group is just short of 40 people most days. It's a relatively small group for the laboratory, which I personally love because I have a lot of early career staff that I get to spend a lot of time with and help to mentor. We focus on a variety of different types of microbes, from bacteria to single-celled algae, and we focus a lot on how we can use those to make products of interest that we use in our everyday world. We use them to make plastic precursors, fuel precursors, other types of polymers, etc. We also house in our group most of the plant or crop sciences work for Bioscience Division, and that's a really nice complement for us because plants have their own microbiome, and so we study a lot how does that microbiome help plants and crops grow and try to improve their resiliency to things like climate change. So we're very focused on energy, environmental, and economic security, and we focus on those things by understanding and manipulating how carbon specifically moves through biological systems, but also other nutrients that I won't have a chance to touch on today. So today I was asked to talk about our plastics work. I have given some versions of some of this talk in past presentations, some of which are out there on the web. And so if you want to ever see a more formalized version of this, I think I gave a Bradbury talk a few years ago, and some of this content is very similar. But let's start with the bad news about plastics, which I think we all know. Even Carol Ann kind of ruefully admitted that we had plastic cups. And this is because plastics are everywhere, and in some cases in a really bad way. So you've probably all seen the stories on the news about how plastics are in our ocean, the giant floating trash mat that is circling around in our ocean, and how this type of litter really affects the ecology in our waters. It has been estimated, and this is an older estimate, but it's estimated that by 2050, which is really not that far away, plastics may take up as much mass on the Earth as fish make up on the Earth. And I also, just this week, heard a statistic, which I will say, but you should know that I have not read this literature directly myself, that the amount of plastics produced in this next year will be on par with the amount of weight or mass of all the humans on Earth. So it's a lot, and so that's a real challenge. A few years ago, plastics were even found in our air. So in addition to hearing about microplastics and whatnot in our waters, this really beautiful study that mostly focused on the Mountain West really identified very small microplastics in soils, as well as just literally floating through floating through our environment, in our winds, depositing in our natural areas, including things like national parks. So they're everywhere. Part of the reason for this is, in some cases, people just throw them out. There's litter, but also even though there's opportunities for recycling for certain types of plastics and a lot of types of plastics, those opportunities are not everywhere in the country. There are certain locations, of course, that don't recycle certain things. And also there's, folks who choose not to recycle. It's a lot of different reasons, but ultimately only about 6% of the recyclable plastics actually get recycled. So there's a huge gap there in just the actual implementation of the opportunity that we have today. Also, as I mentioned, the traditional recycling that's available today, which is called mechanical recycling, grinding up and turning those plastics into something else, is really not applicable to every type of polymer that we have out there. And then also, companies and communities, there's not as much incentive as maybe there could be for getting people to reclaim their plastics. So, you might imagine, just like, let's say you buy your milk in a glass jug, which you can do if you go to like one of the co-ops in Santa Fe, for example, and as long as you're willing to bring back your, you can always buy another glass jar, but there's an incentive, you get a little bit of a buyback if you take your glass jar back. And so companies, it would be really great if there was incentive in some ways for companies to receive back, say maybe their laundry jugs or whatever you want to think of. But those economic incentives are kind of low right now. That all said, plastics are really embedded in our way of life, right? So how many people drove here today? Probably, yeah. Some people probably walked or took public transportation, but lots of people, cars. Part of the whole lightweighting and improving energy efficiency and fuel efficiency in cars, which is really important for other environmental reasons, is due to being able to have different types of plastics instead of all metal in our vehicles. So there's a benefit there, even though there are these challenges. Many of us, if not everyone, have cell phones. Lots of our different types of textiles contain plastics and polymers of these similar types. Now I have a friend who has really committed to only wearing unbleached cotton. And literally his whole wardrobe is, just this really nice light tan color. But he wears almost the same thing every single day, and he's made that commitment, right? But that's not the world that most of us are living in. Fast fashion means that, a lot of our clothes, even if we try to buy high quality clothes, they don't tend to last as long. Our appliances are not made to last as long. Just a lot of items that, as part of our culture, globally even, not just in the U.S., are more disposable, right? And that creates waste, but it is also the reality. I also kind of just like to point out, I mentioned I do have kids. When my husband and I were the classic people of knowing exactly what we were going to do as parents before we became parents. And so we are always going to use cloth diapers, and we will always use glass bottles if I'm not able to nurse for some reason, And I also work full-time. I worked part-time, but nonetheless, I went back to work. It turns out our daycare doesn't take glass bottles because they're afraid if they drop them, it's going to break, and it'll be a hazard to the babies. They really didn't want to handle our cloth diapers, right? And so even, at a reasonably high socioeconomic status with a lot of resources at our disposal, the reality is that we were using disposable diapers at least a fraction of the time. And we were using, plastic bottles at least a fraction of the time. And now my kids are in school, and I'll just send them with, glass. I can send them with glass containers for lunch. Well, I don't know if you've ever seen a kid walking to school, but their backpacks are already huge and already heavy. So am I going to add glass to the weight of their backpack? No, I'm not. You know, I can do stainless steel, and we do a mix of stainless and plastic. So I think it's just an example of like truly how embedded, I mean, even if you just look around the room, plastic is, and how much it has advanced our society in a lot of ways, and it's just not going anywhere. You know, and I think also it's just worth thinking about, you know, for people who are even more time pressed than me and my husband are, or people who are working 2 jobs, don't have access to laundry in their own home, how much harder it is for them potentially to adopt, you know, what we would consider best practices for using renewable or reusable types of products. And so with that in mind, you know, I would say plastics are here to stay, or at least the use of those types of a polymer of that type are here to stay. And so how, you know, if that's the case, what are we gonna do? So we, among plastics, among other types of waste, we really think of plastics as an opportunity and not just a challenge. So it's definitely true that there's loads of plastic going into the atmosphere, or into the environment and the atmosphere, and that is challenges that need to be addressed. But also, we could do something with that, with those plastics. So plastics are primarily made of carbon, just like our fuels are primarily made of carbon, but everything is just arranged differently chemically. And so there are ways to break that down, or we're working on ways to break that down in order to turn it into other types of products, and I'll talk to you a little bit about that today. But I just wanted to kind of frame it out in a slightly bigger picture, because I use this graphic a lot when I describe what our group does, and so I just wanted to share it with you. So on the left is what I call an imbalanced carbon cycle, which includes plastics, and this is where I would say we are today. So a lot of the products that we know and love and use every day are fossil fuel based, including plastics. And that means that we're pulling you know, crude oil, for the most part, from deep within the earth and processing that industrially and turning it into our products. Those resources that are down deep in the earth originally came from biological sources. In some cases, they came from things like biomass, like trees and whatnot. Some cases, they're also from things like algae and other types of organisms from back in, say, the dinosaur days. But the short of it is that crops and other types of organisms that use CO2 and sunlight to grow, photosynthesis, their biomass goes into the earth, and that's the cycle. But the amount of photosynthesis that we have going on in the earth right now, and the methods that we have for taking CO2 out of the atmosphere right now, are not balanced relative to the amount that we're producing, and hence the imbalance that we have with greenhouse gases. What our group and others are really focused on trying to achieve is something that's more on the right-hand side, which is more of a rebalanced carbon cycle. This is not to say that you know, just tomorrow we'll be not using fossil resources anymore, but it is to say that trying to develop methods that can use crops and other types of organisms directly to make the products that we care about every day is really important to us. And for today's conversation, we're really focused on the lower right here, where I've highlighted that we're making fuels and chemicals and products, and they're landing in that pile of dirt, which is meant to represent our landfills. And the idea is to close that circle so that we can recycle those items into other useful products. And so, and you know, I'll note, because I did mention cars, that I'm not focusing on fuels here. It's plastics talk, but there are so many products that come from a barrel of oil, for example, that are not going to be replaced by electrification. So even if we replace all of our cars and trucks and every transportation vehicle by electrification, which, you know, depending on who you talk to, may or may not happen for every type of vehicle, there are always going to be certain chemicals and certain products that you can't electrify. So I can't wear like an electric shirt, right? So that's those are the sorts of chemicals and products that we're also trying to think about in addition to fuels. For those who don't know, sustainable products and this type of thinking that I'm describing to you today are actually part of LANL's mission. So obviously there's the core weapons mission, but we do also have priority areas in biology and biotechnology. 6% of the world's fossil fuel consumption is used to make polymers in industry, and this is a really significant fraction of our emissions. LANL uses all nature of types of polymers and plastics and different types of material for a variety of types of applications. And so this is an area of interest to the lab. The lab also has priority areas in climate science and in the environment. And so this, the sustainable aspect of this is also really relevant to those goals. So I'll also note that there's an increasing number of companies that really care about this issue. This is just a small smattering from actually some time ago. So the Ellen MacArthur Foundation put out a nice report, and it included a number of highlights of companies who have made a variety of commitments focused on the environment as well as decarbonization in plastics, and you can read about those there. There's also the kind of traditional ones like, Patagonia or Chaco or various other more outdoorsy types of companies that will say, oh yeah, we'll send me back your fleece. I'll turn it into a new fleece, things like that. Those folks are still going with their programs and expanding, I would say, in many cases. Not on here is Amazon. Amazon has taken a tackle at a fair bit of their packaging, living in a small mountain town. I do know a couple of people who refuse to use Amazon, but I'm guessing the bulk of people are still ordering from them. I know we are. Okay, so I'm going to talk a little bit about a specific project. This project is called BOTTLE for short. Its long tongue twister of a name is Biooptimized Technologies for Keeping Thermoplastics Out of Landfills and the Environment. Very catchy, very long. And so BOTTLE is the project. And this is actually a multi-institutional consortium. It's funded by the Department of Energy. It's led by the National Renewable Energy Laboratory, which is a sister national lab up in Golden, Colorado. Has a number of other national labs associated with it, as well as a handful of universities. This project is now maybe going on 5 years. I think it's going on in its 5th year. LANL was officially part of the program for the first handful of years, and then since then we have been, they shifted direction slightly, and so we have been wrapping up our work with the last of our funds. And so actually it's a really great time to share with you what we've been up to because we're nearing the end of our involvement in this particular project. But we have a portfolio of plastics programs. This is just one that I like to highlight. Okay, so I showed this and kind of described the idea of a circle or a cycle in that carbon cycle imbalanced graphic just a few slides ago. But this is just another way of showing it, that specifically in the plastics economy, there is a primarily linear aspect to the way these products are used. You get your materials, you make your products, they're used and they're dumped. Some of those items, again, go to the atmosphere, some go into the ocean, some go to a landfill, and again, about 6% are recycled. And what we really want to do is make this more circular, so we are making the products, degrading those products into small building blocks again after they're used, and then ideally, those building blocks can be used directly into making the next set of products. And one thing that I want to highlight that I'm not sure I've defined in any of these slides is that there's different types of recycling. Or cycling, I guess I should say. So recycling, by definition, is taking a product and turning it into the same product again, or turning it into a similar product of the same value. Downcycling is when you take something and you rework it chemically or whatever, you make a new product, but that product doesn't have the same value that the first product did. And then upcycling, everyone's heard of upcycling, like upcycling probably. You take something that's kind of junky, or you can do it yourself, right? You can take some old furniture or whatever, and if you create something that's higher value than what you started with, that's upcycling. So what we call recycling, we say we throw something in our, well, this is number 1, number 2, number 5, we're going to throw in our recycling bin. The products that come out of the process that we call recycling in informal conversation is actually downcycling. So the products that are being made are of less value than the original products that were starting out. And that's part of the reason why companies themselves don't have a real incentive to take back any of the plastics from their used items, because they're not ever going to get back the value out of that polymer that they did the first time around. So part of this project is trying to identify what else can we build with those building blocks that would either be at least as valuable or and maybe identical, so you're truly doing recycling, or even maybe upcycling in certain cases. And overall, because what I'll talk to you a fair bit about today is how difficult it is actually to break down plastics. That's one reason why they live so long in the environment. We also want to design plastics that have the same types of functions that we know and love, but that actually are easier to break down, either in the environment, but also in like a recycling facility. So Bottle has been exploring a range of parallel approaches, and LANL has not been involved in all of these, but so you have plastic waste here on the left, and then there's a variety of different ways that you can break down plastic waste, and they have their advantages and disadvantages, which is why this project decided to tackle a handful of them in parallel. So you can use thermal approaches, so heat types of approaches, heat and chemistry, essentially, electrocatalysis, bio-based catalysis, and then photocatalysis, so light-based catalysis. And the idea then, as I already kind of mentioned, is to take those breakdown products and then develop new ways to turn those into either upcycled materials or what we call infinitely recyclable polymers. And LANL's contributions and piece of this was in the biocatalysis aspect, so the idea of being able to enable biological approaches for breaking down these plastics, and some of that work was nicely highlighted in one of LANL's 1663 articles a couple of years back, and I have not with me, I should have brought stuff with me, but if anyone is interested in reading this or any of our other related climate or environmental types of 1663 articles that we've done recently, I'd be happy to send you the PDFs. So just ask Carol Ann maybe, and I'll send them over to her. Okay, so for the bio-based piece breakdown, it turns out that microbes, so small single-celled organisms, can degrade plastics and eat those products. So based on how much plastic waste there is in the environment, you might not, that might not be intuitive. And that's in part because those microbes have newly developed or evolved that capability. So this is not a capability that microbes have had for a long time. This is a true evolution in the moment type of example where in a waste treatment-ish area in Japan, which is where it was first discovered, there were microbes hanging out in the wastewater there, and there was plastic hanging out in the wastewater there, and the microbes that evolved a path to eating that plastic now had a food source that nobody else did, and that gave them a competitive advantage. So what they do, so my microbes are depicted as these silly green monsters here. So what they do is, and it's not really shown on the left, but they actually secrete or spit out proteins, a type of protein called enzymes. And forgive me if any of you are in a more biological field and I'm talking too simply, but I want to make sure that it's easy for everyone. But anyway, bacteria, they secrete these special proteins called enzymes, and those enzymes then, they attach themselves to the plastic, they break off chunks of the plastic, very microscopic chunks of the plastic, and then the bacteria is able to take those up and then again, use them as a food source. So that's all well and good. Why don't we just use those and the problem is solved? Well, the problem is that they don't work very well. So they work well enough for those bugs to get an extra snack in their day, but they're slow, and they don't work well enough for us to just pluck them out of nature and put them into some industrial process and use them in that way. And that's really common. So for those of us who are in the type of field where we're trying to take enzymes or proteins and improve their function for an actual biotechnology or industrial use, it's not uncommon to have to tweak those enzymes and find faster versions of them, because they, in nature, they're only as fast as they have to be. So also, the other key piece of this is that not only is activity slow, but the scientific process for finding new versions of these that are much faster, that process itself is also slow. The old way is to, look at the enzyme, and we have special ways of understanding what the structure of those enzymes are, and identify different changes, basically single changes that we think would be valuable, and then either making those changes one at a time or maybe a few at a time, and then testing them one by one. And so that way you can imagine could be very slow, especially if there's not a lot of examples of fast enzymes out there, because this whole process is newly evolved. There's just not a lot of examples in nature of which changes to make. So here at LANL, we have developed a couple of different methods for tackling this problem. Both of them can be generically applied to improving enzymes or even whole microbes for different types of processes, but I'm going to just explain these primarily in the context of plastics degradation today. And so the first one is what we call smart microbial cell technology. This won an R&D 100 award. The effort is led by Dr. Ramesh Shah, who is a colleague of mine and a staff member in my group. And this graphic kind of explains the kind of general concept. Excuse me. And so what we're trying to do is develop a way of monitoring the performance of our microbe. So in this case, it would be plastic degradation, the success of degrading plastic by our microbe, by looking at some visual readout. And so in this case, we want to get the cells to glow bright fluorescent green when we hit them with a certain light. And so in this particular case, what this is showing is that this cell is taking in a molecule as food. That molecule is then being transformed into a target molecule, whatever that may be. In our case, it would be a plastic degradation product. That molecule binds to a different protein called a transcription factor, and then when that interacts with DNA, it turns on green fluorescent protein, which glows really bright green. And this is a little example of that here on the right. And so every single one of these dots is not one cell, but a grouping of cells called a colony. And you can see how brightly green they are. Those you can see with the naked eye when you shine a certain light on them. So there's a couple of different ways that we can measure that fluorescence, but it gives us an actual readout to how well our cells are performing. And that gives us an actual readout on, say, if we wanted to make thousands, tens of thousands, or a million different changes to our enzymes of interest for plastic degradation, we can do that and then see which ones glow the brightest green. So this is, again, sort of a more kind of generic example, but in this particular example, we had microbes that were not focused on plastic. They were focused on making a different molecule, like the red dots in the center there. But this particular microbe, as they kept changing it to increase that amount of target molecule, the growth kept getting worse and worse. So now if you're thinking about trying to use this again in industry, you need your cells to grow really well, so you have lots of them, and each cell needs to make a lot of your product. But they were losing one of those features while they are increasing the other feature. So they went through a process, it's called adaptive evolution, and it's basically allowing the cells to generate a bunch of their own mutations. And then we added what we call this biosensor, which is this little gray X there that actually senses the presence of that target molecule. And when we did that, we were able to physically pull out the cells that both grew well and still produced a lot of that target molecule. And that turned out to be a better method than any of the previous methods alone. And so this same type of approach can be used for plastics. It's a little bit trickier because now the enzyme that's making your target is outside because it's stuck to the plastic. But it's a very similar idea where now you have each cell is making a different version of the enzyme, and that enzyme is getting out of the cell, interacting with the plastics, creating degradation products which go back into the cell, and then the more of those there are, the brighter green that cell is. And so now you know that particular enzyme variant is more effective at doing the degradation than maybe some of your other variants are. And you can do this, again, you can look at this in multiple ways, but just like Ramesh was holding up that plate and they show they're all bright green, sometimes you hold up the plate and some are very dim, some are super bright, some are in the middle. And so you can just scoop up those super bright ones, then analyze them more, and then add more mutations on top of those. So that's an approach that we've been taking. Every time we want to do something like this, we have to basically customize that transcription factor, that gray X there in the middle of the cell. And so we have customized the transcription factor for, I think, 1 or 2 degradations of products at this point, and have been working on using those for creating variations of some of those breakdown enzymes, and then trying to screen for the ones that are the most improved. We ended up tackling nylon for this problem instead of nylon would be more like your shirts, right, versus like your water bottle or your Coke bottle. And polyethylene terephthalate is the other plastic, and that's the one that's known the most about for biological breakdown. And so we started with a really hard problem with this. So I don't think they're quite ready to publish this work yet, but it is coming along really nicely. In the meantime, we had the second method that is also based off of this kind of bright green concept, but is just designed slightly differently. And this particular method has been used to tackle actually kind of your classic water or soda bottle that you might buy or someone might buy in the store. And so here, not only are we caring about how well does the enzyme actually do its chemistry and break down the plastic itself, but they also had this cool idea, and this work is led by Hal Nguyen and her team in Bio Division. We collaborated with her on this. And so not only are we focused on activity, but also these plastics are more amenable to breakdown if you do the reaction at high temperature. And just like us, we kind of wilt in really high temperatures, and so do enzymes. So the enzymes that we're working with, at least, were originally discovered in organisms that are most happy at sort of what we would consider room temperature, and we're really looking at like higher industrial temperatures, more as 70 to 80 degrees centigrade. So they also wanted to at the same time try to improve the temperature tolerance of these enzymes, and then similarly they want to make sure that whatever enzymes they are pulling out are ones that are easy to make a lot of. So if you're imagining that you're going to fill a whole tank with some of these enzymes one day to break down a lot of plastic waste, you need, again, to be able to make a large amount of the enzymes. And sometimes when you do a lot of these changes to the proteins, the cell stops being able to make very much of them, or they stop folding properly, and that kind of is very counter to what you're trying to ultimately get done. So they basically created what they call a co-screening assay. They're picking enzymes that meet these 3 criteria normally you kind of have to do one at a time. And I won't get into the major details, but the short of it is that they took enzymes that were already out there in the literature. They made a lot of different variations all at once, a lot of mixing and matching of the different parts of the enzyme, and then they put them through their screen, pulled out the ones that they felt had all of these 3 criteria, And then they tested them. After they tested them in the laboratory, they sent them to a collaborator in the UK who tested them in bioreactors. In this case, they tested them at 65 degrees centigrade, so not quite at that 70, 80, but still much higher than room temperature. And then they also tested them on actual, imagine a hole punch and hole punching out little circles of your soda bottle. So actual, what we call coupons, but they're little hole punch circles. Put those right in the reactor and then put in a pile of the enzyme and then compared it to other enzymes that were already out there in the literature. And the Hames one, which was notably named by one of our postdocs, is the best one in this comparison. So we're really excited about this work. So the LCC-ICCG is a literature-based. It's an enzyme that someone else discovered and published in the literature. PHL7 wild type, which is the kind of navy, dark, almost black color, is also a literature-based one. And then LCC-LANL obviously is one of ours, and then PHL7 Hames is one of ours. So we're really excited about these data. This team's actually preparing a package for an R&D 100 award this year, and so we're hoping that they'll get some recognition for that. I didn't think of it until just now, but I have this really cool photo. They have this really cool photo where they took like a plastic strawberry container from Smith's, and they treated it with their enzymes just on the bench, and like within a day or two, it's just it's just in this goo, like it's totally gone. And so yeah, so again, maybe I can send you guys that photo if anyone wants to see it, but it's a pretty compelling picture. Some of the things that we're kind of working, we focused a lot on the plastics work on this, again, the type of polymer that forms your soda or water bottles. And again, I mentioned that, some of the next things we wanted to do is nylon. We've already started working on nylon. Some of the harder problems are things like polyurethanes, foams. That type of chemistry is very different, very difficult to handle biologically. And then also, if you think about it, almost every product you have is not just one plastic. Even just one, like, simple, Coke or water bottle is the middle of it, where you can kind of squeeze it. It's one polymer, or one version of a polymer. The base, where it's more firm. Now, when you see the kids doing the flip bottle thing, the base is more firm. That's different. The cap is different. The label's different. So, and that's true for a lot of different things, and they may be just in different areas of the product, but they can also be layered. And so this is one place where the biological approaches could potentially really come into play, because if you have an enzyme that does all of them, as long as their conditions are compatible with each other, you can make a mix, and each enzyme will go to its part of the product. So these are some of the areas that we're moving towards. I think we have there's a funding call out that we're thinking of tackling the polyurethane problem, and then just a couple more. So just this is sort of back to this idea of not only do we want to break down the products, but we also want to get microbes to uptake those products and use them to make new products. So whether they're making new monomers for the polymers of the future, or whether they're making monomers for the exact same type of product, depends a little bit on the application space. But the idea is that we have a lot of experience, and this consortium also has a lot of experience with taking microbes and engineering them to take up unusual molecules and make stuff with them. And so we have, in other cases, helped identify the best versions of those microbes using the same type of technology. So now, instead of focusing on plastic degradation products, you can use the same types of screening to say, I want this microbe to make X type of chemical out of that degradation product. And now our transcription factor can look for that particular chemical and glow bright green. So that's some of the other efforts that we've had going. And then this is really recent, so I just wanted to highlight this. This is a review article, and I haven't had a chance to read it very carefully myself, so I couldn't say how super technical it is or not. But this came out of our group just this week, and we were able to get the cover art for the journal. So I thought it would be fun to recognize Jessica and Babs for this, as well as Gansham. So in the thought process for designing polymers of the future, so let's say we want to get away from the plastics that we have today, and we want to keep the function, keep the convenience, but improve degradability. And ideally even design it so that maybe it really only lasts, the lifetime really is as long as the function or the performance needs to be, and then maybe the natural, even the environmental breakdown is enhanced. And so there's a number of different pieces that you have to consider going into that, and this kind of tetrahedron on the left highlights that a little bit, that you have the chemical structure, and of the chemical itself, the polymer itself, how that polymer is processed and actually turned into a material is another feature that's important, and both of those feed into the types of properties that then the material has, and that affects the performance. And so, and performance, as I said, includes everything from, how does the user use it, to how does it affect the environment, ideally not as badly as the plastics we have today. So this review highlights those things. Babs Maroney is the PI of this work. It's been going on for a while. Jessica Lalonde, whose photo is a little small here, she was a graduate student with Babs. She was a graduate student at Duke. Babs was on her committee, and then she's just started here as a postdoc. We're excited to have her back. And then Gansham is a collaborator with us at LANL. And so I'll just, a lot of people to thank. I'll just highlight, since I spent most of the time on the bottle and NREL, Greg Beckham is the PI, and the work that the polymer chemistry project came out of previously funded LDRD work. And with that, I'll say thank you. And I don't know how the timing was, but
probably aren't getting off scot-free here.
that's fine. Yeah.
And I know it's 1 minute.
Oh, is it? Well, we have 5 minutes, so.
But how do you want to do that with the slide?
If somebody puts up their hand, I'll go over to them.
Okay.
She just needs to repeat the question.
Okay. So does anyone have a question?
Sue Ellen.
What would you say to someone
Wait.
burning plastic in their garbage?
Why
Why is that not a good idea?
So the question is what would you say to someone who's burning plastic in their garbage? I depend on that someone because my parents still do this in Pennsylvania. I think, obviously it's not a good idea. The reason it's not a good idea is because burning plastics creates CO2 and it goes into the atmosphere. So that's definitely not what we want. I think it's recognized generally that burning trash in general is not really ideal way to get rid of your waste. And I say that, and so that's what I would recommend and advocate for, is not doing that. I also think it's important to recognize that, again, there's some fraction of people that is their path to getting rid of their trash, right? And so in my mind, creating products then that it's maybe better to throw in a dirt pile next to your trash pile and they're going to naturally compost would be a better product to have, and encourage people to do that instead of burning. But, I think this is the balance of what do we wish everyone would do versus what is practical for everyone to do. And then in some cases, what, people are just going to do what's easy. And so I think, that's a hard question, the social side of it. Yeah. Yes.
Some of us in the room are knowledgeable about the bureaucratic challenges of working at LANL.
Mm-hmm.
I'm just wondering if you could ever see your division spinning off and becoming an independent organization, thereby reducing overhead costs and investing more in this vital research?
Yeah, thank you for that question. So, to repeat the question, there's a lot of bureaucracy at LANL, and have we considered, or would it be beneficial to spin off this work or our division to be an independent entity and reduce overhead? Yes, there's a lot of bureaucracy at LANL. Even though we're a small division, though, we have received a lot of support from LANL, from pretty high up in the management, especially around this type of work. And so I'm not thinking seriously about carving out our entire division to be an independent entity. I will say, however, that there are lots of examples of, especially when a technology has been patented and looks very promising, of getting external independent industry investment and doing either the PI leaving for an entrepreneurial leave and starting a company, or actually just leaving to start a company. And so there are examples of that, of course, quite a few out of bioscience. And out of Bottle, actually, I know a couple of the other PIs have started a company with some of their other technology. We have the patents are for this work, some of the work that Hal has done. The second piece that I talked about, we do have patent applications in. We're waiting for them to be granted. And so I think, you know, if the company says that they want to come and license that effort and pay for it and start actually using it industrially, we would be very excited to see that type of success. For the transcription factors, the sort of like the cells that glow green, we have other examples of making chemical replacements or chemicals that have moved out of the R&D space and are now moving into other projects that are heavily supported by industry to try to commercialize. So we kind of sit on that edge, but when people ask me personally, do you want to start a company? I'm like, I don't know. Sounds like even more work, but yeah.
Hi, I'm sorry I have to get way back to the basics.
Sure.
The reason that this works is because plastic is carbon-based, and life is carbon-based, so the enzymes are carbon-based,
Mm-hmm.
and that's the basis of this. So way back in your initial slides, you talked about choosing enzymes that work on plastic. Can you just briefly restate that?
Yeah, I can try. So, just to try to make sure I understand your question, I guess. So yes, it's true, life is based in carbon. Our enzymes are also, their backbone is made of carbon. Actually, the way they do their chemistry involves other atoms, often things like nitrogens and oxygens and other elements of life. The plastic itself is, and in fact, maybe I can kind of point to it, even though it's a little small here. So that there's the O's for oxygen, and then the line in the center that's kind of wiggly is carbon. Those are carbons that are linked. They just don't highlight the C's. And so the reason why certain plastics are attackable by enzymes is actually because of the oxygen. So if this was only carbon, it would actually be harder. So I'm not sure I'm answering your question, but the short of it is that there are molecules that are similar enough to these that nature already has some enzymes that are kind of used to doing that work, and then when they're exposed to these in the environment, they tweak themselves a little bit to be able to do it.
Thank you. I think that's it. I understand what was happening. I just didn't understand how.
Yeah. Okay. So if you think of, I didn't really show that graphically. If you were to really zoom in, we sometimes draw enzymes as little Pac-Mans. And the reason for that is because they do have, the mouth of the Pac-Man would be what we would call the active site. And the enzymes have a way of pulling basically very tiny pieces of the polymer from the whole, like if you're really zoomed in to the plastic bottle, then it's actually kind of more of a rough surface, and they're able to connect themselves to that and pull on a piece of the fiber and then chomp at it in certain locations. And then depending on how good it is, it may stay stuck, or it might have to fall off and reattach and then chomp again. And so you have to kind of think of them as little chompers. Yeah. Does that help? Okay. Yeah. Oh, did you? Well, there's walk past one. Oh, yeah.
How innocuous are the breakdown products of the enzymatic reaction?
Yeah, good question. So I don't have that in here. They're pretty innocuous. I mean, I think our goal is to, our vision really would be to do this in these particular reactions in a reactor where those breakdown products are contained. Also because, again, we want to keep that carbon and turn it into something else. And so if we were talking about releasing these enzymes into the environment, it might solve our waste problem, but it doesn't allow us to get back that carbon. So, yeah. One of the breakdown products is actually is an already established food source for some microbes, so they would just eat it and use it. Yeah, go ahead.
So, how far in the future do you see your research being used widespread?
Yeah, how long? This is always a good question. I think I was asked this question 5 years ago, and I might have said in 10 years, and it's always the ticking clock. I mean, I think if we could see some version of this happening, maybe not in every city, but in some cities, by the time I retire, and that's like maybe in 15 years, I think in the 10-ish plus or minus 10, 15 year type of time frame, I think is very reasonable. There are companies in Europe who are developing their own versions of some of these enzymes, and I don't think they're making money yet, but they're still well alive. Again, there's at least one spinoff from this one project from the bottle project. There's other companies focused on this, so I think there's also going to be other approaches. So I mentioned that bottle was focused on some of these other ways of doing degradation using other types of chemistry, not bio. And some of those actually have kicked off to be like even more successful near term, which is in part why LANL has peeled off for this project in the last couple of years. And so I think there's going to be additional recycling options and definitely better polymers coming out for products, probably even sooner than that. So, yeah.
You mentioned, you keep mentioning the ease of and speed of degradation. The opposite end, is there any research at all on increasing heat stabilities to make the plastics more durable so that they could be reused in times, kind of like glass isn't?
Yeah, I actually really like that idea. I have not heard people talking about that. I don't know if it's because we always couch this conversation in the waste problem, and so, but yeah, I mean that kind of just gets if we can get people away from the use it one time kind of idea. Yeah, I haven't, but maybe there are people who are working on that, and I'm just not aware of it. Yeah, it's like kind of moving away from plastic baggies to the reusable, pack your sandwich in the slightly thicker bag type of idea. Yeah. Did you want to have? Okay.
Two things. I do want to say yes, I know these are plastic, and I put them in my dishwasher and wash them every time.
Yes. Yeah.
We've had it for at least 10 years. But the other thing is something popped up on my computer. I don't even know where I was, but it said something that I didn't know. So those plastic bottles which I actually don't buy that water comes in.
Mm-hmm.
It said you should not squish them. You should leave them whole to recycle because it was better. Is that true?
I don't know the answer to that specific question. We still tend to kind of squeeze ours this way, at least, like this way, just so that you can get more in your bin. But yeah, I don't know the answer to that. I think that would be a good question to know the answer to. Yeah. Yeah. You have to look it up.
I'll
Yeah.
look for that source, but if you know how
Yeah. Yeah.
and you probably are all doing this. I said,
Oh, sure.
And they said you shouldn't do anything, you should just put the lid on and put it in the
Yes.
recycle.
Yeah. Yep.
You've been doing that wrong.
Yep. Well, yeah, and I am aware, there used to be this sort of guidance that, they can't do the lids. Take off the lid for everything. And I think the latest paper that I saw was if the lid came with the thing, leave it with the thing. Yeah. So do that. Okay.
I can't thank you enough.
Yeah. Thank you. Thank you very much.